I'm Louise Robson. I'm the chair of the Education Committee at the Physiological Society, and it's my pleasure to introduce the recipient for 2010 of the Joan Mott Prize Lecture. The Joan Mott Prize Lecture started in 1996, and it's normally awarded every 2 years to a female academic who's really made a massive difference to physiology, and not just that, someone that can act as a great role model for young female researchers who are going to aspire to be in that position in the future. And this year's winner is Professor Dame Kay Davies. It's absolutely fantastic that we've got her here today. She's the head of department at Oxford, holds numerous other very important positions, and her research is about muscular and neurological diseases and the genetic basis of those diseases, particular muscular dystrophy, and it's my great pleasure to introduce her.
It's a great pleasure, and I'd like to thank the organising committee for proposing that I have the opportunity to tell you about muscular dystrophy today. I'll start by saying I started off as a chemist, as Roger Chen did, but I haven't quite achieved that status. But I think what we have done is make a big difference to muscular dystrophy, and that's the story I'd like to tell you. And it has required a lot of collaboration with a lot of international people as well as people nationally. And it also has required a lot of male mentorship. And so I think for women to succeed, since I just have one note on that, we both need female and male mentorship throughout our careers to continually encourage us to keep going. But science is fun, and you'll see how this particular project has turned in various ways as we've gone through from isolating the gene all the way through to thinking about therapies. Just one word on Joan Mott. Our paths did cross because as a young chemist who didn't like chemistry very much in 1975, I applied to do a studentship in biochemistry with Ian Walker, and it was Wolfson College, and Joan Mott was a founding fellow of Wolfson College, that encouraged me not to go into teaching but to think about doing a PhD, and for that I am forever grateful. That was the first bit of important mentoring. And secondly, I was then given a postdoctoral fellowship at Wolfson College, the first Guy Newton Fellowship. And again, Joan Mott played a part in that. So this is a tribute to Joan in more than one way. So let me now introduce you to the disease I'm going to talk about. It's a very tragic progressive muscle disorder. It is the most common genetic form of muscular dystrophy. It's Duchenne muscular dystrophy, first described by Duchenne. Its prevalence is about 1 in 3,000, so it's the second most common genetic disease after cystic fibrosis. As I've said, there's no curative treatment, although just pure management and respiratory care towards the end of their lives has now extended the life expectancy from about 20 to about 30. Having said that, most of these boys, in fact, the clinical criteria dictate they're all wheelchair-bound by the age of 12. And if they are wheelchair-bound a bit later, then they're thought to suffer the milder form of the disease, and some of them, as I'll show you in a moment, can not demonstrate symptoms until their 40s or 50s. That's called Becker muscular dystrophy because Becker first characterized the milder form of the disease. So they're not diagnosed, though, until about the age of 5. And when I first started working on this disease in 1980, there were many families with multiple affected SIBs. And in fact, there still are. And these are just 3 brothers, just showing you the progression. This boy at 2 years, if you didn't know that there was muscular dystrophy in the family, he looks relatively normal. And these are his shots taken of his brothers later on, where you can see the muscle wasting and the scoliosis, the curvature of the spine that plagues them later on in the disease when they go into a wheelchair. And I was fascinated by this because this particular disease has a very high new mutation rate. So if there's any disease that needs a therapy, this is it, because unless we can screen every pregnancy, DMD will be with us in every population in the world. And we don't diagnose it at birth because there's nothing we can do about it, and so this sort of problem at the moment will recur until we can find that effective treatment. So, high new mutation rate. Why is that? Well, it turns out to be quite a remarkable gene. It's the biggest gene in the human genome. It covers 2.7 megabases of DNA sequence, and if you just compare that to the factor VIII gene, which only covers 200 kb, a relatively large messenger RNA of 9 kb, the DMD gene, which encodes a protein called dystrophin because lack of it causes dystrophy, is a 14 kb messenger RNA. And there are 79 exons, and I'll come back to this at the end of my talk, and sometimes the introns are 120 kb or more between each of those exons, which on average are only 150 base pairs long. It encodes a very large, but not the largest protein in muscle, a 427 kilodalton structural protein. And amazingly, this isn't just a large target for mutation in that 2.7. There are hotspots for mutation in this gene in two particular regions, and 65% of these patients actually have bits of their gene missing. So this is a muscle biopsy showing the normal individual with dystrophin localized at the sarcolemma there, and in most DMD patients you get complete absence of the protein, as you see here. Occasionally you'll get a revertant fiber where there is a small deletion in this particular patient's gene, and nature itself skips exons, puts the protein back in frame, and produces a shortened protein, which, as you can see, is still functional here in the sense that it can localize to the membrane. In the milder form of the disease, you either get less of the dystrophin or it doesn't bind so well to the membrane, and this is just a typical BMD patient showing the much decreased level of a shortened dystrophin at the sarcolemma. So Lou Kunkel first identified dystrophin back in 1986, and Tony Monaco, who's then in his group, produced the open reading frame rule, whereby if you had an outer frame deletion and you didn't produce any dystrophin, you got DMD. If you had an in-frame deletion, depending on where that in-frame deletion was, you could have a shorter protein that was partially functional. So what does dystrophin do? As I said, it's a structural protein, and if it itself actually doesn't lie across the membrane. It forms an important link between the internal cytoskeleton with F-actin here by this N-terminal domain. There are then spectrin-like repeats, a rod domain here, and then this very important C-terminus that binds this complex of protein, the dystrophin-associated protein complex, that links then to the extracellular matrix. And this is work of Kevin Campbell's group and Ozawa's group in Japan. And if that theory is right, of course, if you get mutations in any of these other muscle-specific proteins that are encoded by autosomal genes, you would expect to get a similar phenotype. And indeed you do. There are patients with autosomal recessive muscular dystrophies that have mutations in these genes here. So if you take dystrophin away, you destroy this link between the extracellular matrix and the internal cytoskeleton, and then you lose that complex, and the sarcolemma becomes very much more fragile, and you get the manifestations of the disease. And that's why well, that's not the only reason. I'll come back to that in a minute. When you get more and more exercise, when these children are born, they have very high creatine kinase levels, indicative of muscle damage, but it's not sufficient for them to show any impairment physically. It's only when they have more and more exercise and then they start to climb stairs that this particular disease becomes manifest. So, this is a disease that really needs a therapy. This is a disease that perhaps is one of the worst to produce a therapy for because it's a structural protein. And experiments in mice, and more recently by Francesca Montoni's group in man, demonstrate that you need at least 20% of that protein in order to get to any clinical benefit. You need to target all muscles because most of these patients, by the time they're 18, have an abnormal ECG. You've got to get it to the skeletal muscle and the diaphragm. And of course, muscle can be up to 40% of a person's body mass. So if you're trying to put it in with a virus, you've got to think about avoiding the immune response, which is also quite a challenge. And of course, you'll need a lifelong treatment because you will have to replace dystrophin all of the time. So I'm going to give you a quick review of what therapies there are available, and then I'm going to concentrate mainly on two. And I'm mainly on that too in an unbiased way, she says, because one of these, exon skipping, is in the clinic, and this is also now just going into a phase 1 clinical trial. I will also declare that I have a conflict of interest here because I set up a company because no big pharma would at this stage do anything to do with DMD. So we set up a company to try and tackle it, and now the big pharma are moving into DMD, which is good news. So there are growth factors. You can increase the amount of muscle mass. Myostatin inhibitors, IGF-1, some of those have been through clinical trials, but they're not very effective, and I'm not going to say anything more. Stem cells, I have to tell you that there is a clinical trial by Giulio Cossa's group in Italy that is delivering stem cells systemically to DMD patients. I'll make no other comment except that I think that might give us some clues, but it certainly won't lead to a cure. And experiments recently from Tom Rando's group indicate that even if some of these cells, these stem cells, migrate to the site of damage in DMD, they then don't differentiate into muscle cells because you need the right environment in order to stimulate them to differentiate into myoblasts and myotubes. One of the most promising clinical trials, and actually even 2 months ago I would have stood here and said, if you could read through the stop codons in the 6% of patients that have DMD, you'd be able to get a full-length protein dystrophin out. And that's been a strategy that PTC Therapeutics have used. They developed a compound called PTC124 that you may have seen in the literature that suppressed the stop codon. Unfortunately, this turned out not to be effective clinically in human trials. And in discussion afterwards, I can tell you perhaps why some of that may have happened. But there are other agents that can suppress stop codons, so maybe other agents that come along. What about gene therapy using viruses? This has also been in clinical trial, particularly by Jerry Mundell's group. The problem here is you've got a 14 kb messenger RNA, and most of these viruses only take up to 7 kilobase pairs of sequence. Remarkably, the late Sarah Bundy sent me DNA from a patient, and these are the gentleman. This is him after he's just come out of the army. This is him at age 45. He's just climbed up that cliff, but you can see he needed the aid of a walking stick, and he's got the characteristic gait of Becker muscular dystrophy here in this picture. And amazingly, this patient has the largest mutation that's ever been described in the literature. 50% of his gene is missing. Well, it's not only amazing, it's also extremely fortunate, because he's missing that spectrin-like repeat of the protein. So he still has, this is his mutation here, schematically drawn, he still has the binding partner that attaches the internal cytoskeleton via F-actin, and he still has all the carboxy-terminal domain intact, which binds that complex at the membrane. And he's managed to dispense with most, but not all, of this region here. And since then, other patients have been described, and Jeff Chamberlain and colleagues have made microdystrophin, we call his gene a mini-dystrophin, in order to get these things into viruses. Oh, and I didn't include it. So using that type of approach in AAV vectors, Jerry Mundell has actually now completed a clinical trial. There is an immune response to the vector, which you'd expect, but he also observed an immune response to dystrophin itself, which is a bit of a surprise. And so if these patients have never seen dystrophin, and you're going to think about a long life treatment, you've got to be a little bit more creative than perhaps just putting dystrophin back in, and I'll come back to that at the end. So we thought that maybe there might be a better pharmacological way of treating this disease where we wouldn't have an immune response that might be mutation independent, that might target all muscles. And I was particularly fortunate because I was then in the Institute of Molecular Medicine, which was working on, with David Weatherall, who was working on upregulating fetal hemoglobin to replace adult beta globin, which is mutated in thalassemia patients. And Don Love in my lab in 1989 identified a sequence which turned out not to be on the X chromosome, but was remarkably similar to the dystrophin molecule which is missing in DMD patients. So we called it Utrophin because it's ubiquitously expressed. You can find it everywhere. And it is also expressed in adult muscle. I'll come to that in a minute. This is, again, demonstrating that it has identical, 97% similar sequences at the vital end that binds that N-terminal F-actin and here that carboxy-terminal complex, the dystrophin-associated protein complex. It's not as highly conserved in this spectrin repeat domain here, and that we know is dispensable to a certain extent because of the patient I've just shown you that had 50% of his gene missing. I should say, actually, I went to his 65th birthday and he died of cardiac failure, age 73. So certainly 50% of your gene missing, if it's the right position and the right deletion here, is certainly compatible with a good lifestyle. So anyway, coming back to utrophin, where is it localized if nature itself has another gene that is so similar to dystrophin and it's expressed in muscle? Where is it expressed? Here's dystrophin again at the sarcolemma, and here's utrophin. You can see that this is not at the sarcolemma but at the neuromuscular junction. And here I'm just showing you the data in mice, and the MDX is the mouse model for DMD. It's got a stop codon in exon 23. And it doesn't produce dystrophin. And it does have the occasional revertant fibre, which I've done here, where the exon 23 is skipped to put it back in frame. So you can see in the MDX, utrophin starts to spread across the sarcolemma. In fact, what is happening here is the utrophin protein is stabilized rather than transcriptionally upregulated in the absence of dystrophin. So that was the first clue that utrophin might be able to, in certain circumstances, replace dystrophin. And so our hypothesis was that if we could increase the levels of utrophin in the MDX mouse, we might be able to cure the mouse, whatever curing the mouse means. And I must say that if I brought the MDX mouse into this lecture theatre, you would not know it suffered from muscular dystrophy, because mice can regenerate their muscles extremely efficiently. You don't get the connective tissue invasion, and it's able to regenerate in a way that man does not. It also, of course, stabilizes utrophin, and that partly compensates, and I'll come back to that in a moment. So there was another clue that this might be a possibility. If you look at patients, and this is antibodies that were supplied very early on in this research, but gives me an opportunity to acknowledge Glenn Morris, who produced these monoclonal antibodies. There's dystrophin that I've shown you before. We called it the DMD-like protein, utrophin, at the beginning, only localized at the neuromuscular junction. But in Duchenne patients, you start to get spreading already of utrophin across the membrane. And indeed, the amount of utrophin that is that you see at the membrane is very variable between patients. It's obviously not enough to cure them, but a recent paper, well, not so recent now, 4 years ago, by a Greek group, demonstrated that the amount which you naturally upregulated appeared to correlate with the age at which you went into a wheelchair. So the more you expressed utrophin, the later you went into a wheelchair. Although this was a very small sample, it was at least indicative that in man this might be a therapeutic possibility. Perhaps more compelling is if you look at human fetal development. And this is what's demonstrated here. This is Caroline Suri's work in collaboration with us that was also confirmed by Fernando Tomi's group in Paris. This is dystrophin in early development through to a 9-month infant. You can see very early in development both utrophin and dystrophin are expressed exactly the same place at the membrane. And then as you go through development, the amount of utrophin, the dystrophin at the membrane increases and the amount of utrophin decreases until at birth you've only got utrophin at the NMJ. So these are virtually identical proteins in the domains that matter, but it's the regulatory control of these two proteins that's different. And that's why you get utrophin expressed where we see it and where you get dystrophin at the sarcolemma. So that's the hypothesis. If we increase utrophin in the mouse, what happens? And so we've published this a long time ago now, but just very quickly, you can see the necrosis, centrally nucleated fibers that are so typical of regenerating muscle in this H&E staining of MDX. And this is an MDX utrophin transgenic, which is expressing utrophin under the control of the actin promoter. So quite early on in development, and you can see now that much of that necrosis has gone, and I think there's one muscle fibre that's got a centrally nucleated fibre here, but I can't find it. Yes, I can, it's there. So you get a dramatic increase. In fact, physiologically, we cannot tell the difference, and I don't have time, it's all published, between the normal mouse and a Utrophin upregulated MDX transgenic. Further, perhaps evidence that Utrophin is playing this reciprocal role, dystrophin, is that, as I said to you, MDX is very mildly affected. What happens if you take Utrophin away as well? Well, if you take Utrophin away on its own, you get a virtually normal mouse. And that was an incredible result, which just goes to show that you can never predict what science is going to do. It's a highly conserved sequence in evolution, and yet you can get rid of Utrophin, and there are one or two problems with your skin fibroblasts, but other than that, there's nothing. If in the MDX, however, you take Utrophin away, you get a very severe progressive muscular dystrophy, very much like the clinical course that you see in DMD. And this is just, this work was done by Anna Deckening in the lab. The mice are smaller than their litter mates, and you can see the kyphosis, the curvature of the spine, it's very similar to what I showed you in the patient in the very early slides. These mice last for about 20 weeks before we actually have to humanely kill them. So, double mutant mice are severely affected. That demonstrates again that Utrophin is very important for the survival of muscle in the MDX mouse. And this provides us with a rapid means to test some of those gene therapy protocols that I was talking about earlier. And I'll come back to that at the end of my talk when I talk about exon skipping. So, Utrophin upregulation then, we know from hints from the human, from the mouse experiments, that all we have to do is increase the levels of Utrophin. That's now established. How much Utrophin do we need? And the answer is about 3 to 4 times the wild-type levels that you would normally see in muscle. Now, what I haven't told you is that if you look in your liver and your kidney, you've got about 10-fold more than what is normally expressed in muscle. So nature, again, in the liver and the kidney has the ability of producing lots of Utrophin. I have no idea what it's doing, but it does. And so the regulatory machinery must be there to produce high levels of Utrophin. How early in life would we have to administer a drug in order to have an effect? And the answer to that is exactly the same as with dystrophin. The earlier the better. We would only arrest the disease, we can't reverse it if we put Utrophin back in, and that's going to be a problem when it comes to clinical trials and trying to work out whether they work. Does increased Utrophin throughout the body have any side effects? Now, this is a crucial experiment because no drug is ever going to be tissue-specific. Aspirin's not tissue-specific. That's the reason it's so useful for so many things. But the key result is how toxic is it to have lots of Utrophin at a therapeutic level that would give you rescue in muscle in your brain, in your spleen, in your liver, and everywhere else. And if you do that experiment, which Rosie Fisher did through transgenic experiments in the mouse, then you don't get any pathology at all in any other tissue. So that was good news. So the only challenge then was to find a drug that would increase the levels of Utrophin. And Utrophin, that is, like dystrophin, is a huge gene. So it's only 1.3 megabases as opposed to 2.7 megabases. But unfortunately for us, the promoters are spread out of 120 kilobase pairs of DNA. And it has 2 promoters that we know of, 1A and 2A exons for promoter A, with an ATG here, with an ATG there. DU is an enhancer element, and promoter B produces Utrophin B, which has a completely separate first exon, which splices into exon 3. So are there any features of the particular regulatory sequence that would, first of all, tell us why Utrophin is at the neuromuscular junction and dystrophin is not? Well, if you look at promoter B, that's only expressed in endothelial cells. It's not expressed in muscle at all. I'm not going to talk any more about it. For the aficionados, it's got everything you would expect of an endothelial promoter. And I'm going to move swiftly on. If you look at promoter A, however, you will get what you might expect of a protein that is localized at the neuromuscular junction. And I'll see if I can read it off here. I'll just have to say, because I think it's TTCCGG, the N box that Changeux characterized as being very important for localizing the acetylcholine receptor at the neuromuscular junction, is present in the 5' end of utrophin. And that is why it's localized to there. It's the regulatory sequence here and this signaling cascade that makes utrophin be synthesized just under the synapse. If you mutate the end box, then you'll get utrophin expressed at the sarcolemma. So if you can interfere with any of these pathways that feed into this end box, you can potentially disrupt its influence and get some of the utrophin spreading onto the sarcolemma. Now, just in case I forget to say it, of course, the end box is a very important sequence. I've already told you it controls the acetylcholine receptor. So if you fiddle around too much with that, you might end up by curing one disease and creating another. Very fortunately, all of the drugs that we've looked so far, this pathway here that feeds into the utrophin promoter is separate from the signaling pathway that dictates the end box in the acetylcholine receptor. So it doesn't look as if we're going to be in danger of creating another disease while curing the other one. We know everything about what this does, so what we did was to put a luciferase gene in frame in this region of the gene so that we could monitor the expression of promoter A, which we know is so important for its expression in muscle. So we screened 5,000 compounds. We assayed the utrophin messenger RNA levels, and we looked for compounds that would upregulate the gene 4 to 5-fold in the test tube and in the micromolar range. Whoops, wrong thing. Right. I'm not going to go through that structure. And this is just one example. The ones that upregulate a small amount, first of all, upregulate at the neuromuscular junction. There's the control, there's treated with the drug. And gradually, after time, that spreads across the sarcolemma. This is the percentage force drop. That's dose dependent. And this is the level of utrophin. I don't have time to go into detail, but suffice it to say that we have one drug which does increase the levels of utrophin, that does all of these things that you can read faster than I can. It reduces the muscle degeneration and necrosis. It's drug-like. In other words, there are no toxicities in animal models, in large animals. It's easy to synthesize, so we can scale it up. And it appears so far in all of the dogs, pigs, and mice to be fairly safe. So the situation for the utrophin program at the moment is that lots of biotechnology companies are also trying this strategy, which is good news. I hope we win, but I don't care who does. We can do it in the mouse. We've got a candidate preclinical drug, and these trials are now ongoing. Bimaran Pharmaceuticals now made a partnership with Summit PLC and are now have taken this into phase 1, which is just safety trials in normals, and those data will come out in about a month's time, and hopefully by the end of this year we'll start to go into patients to see whether we can increase the levels of utrophin in DMD patients. So the final bit of my talk will be about exon skipping. And this is a remarkable and very neat little strategy that wasn't developed by my group, but by the Dutch group originally, but has been taken on by many groups around the world. And that is to take advantage of the fact that you can miss these exons out and you can put this protein back in frame. This work was done by a French postdoc, which is why every now and again the bits of the slide will come out in French. Now, so the principle is, supposing, for example, you had a deletion of exon 44, you can see that if you joined the protein back together again, you would be out of frame because these ends here are not in frame. But you can also see if you took out another exon, exon 45, if you skipped exon 45, then you'd put the protein back in frame and you'd get a partially functional dystrophin because you'd have a Becker phenotype, but it would enormously improve your quality of life. And that's the principle of exon skipping. So how can we do that? Well, we know an awful lot these days, fortunately, about what sequences in the genome govern exon skipping. And there are splicers, there are enhancers, they occur within the gene, they occur at the donor and acceptor splice sites. And so by designing antisense oligonucleotides to various parts of the gene here, which will hide this machinery, you can induce the exon skip. So where is the world in exon skipping? There are 2 clinical trials which have been completed. The first was in Leiden by ProSensor, and they used 2-O-methyl antisense oligonucleotides, and in some cases they got 64 to 97% of myofibers that expressed dystrophin. But this is going into the EDB muscle by direct injection. And that has now gone into a systemic trial, which I'll come back to in a minute. The MEDEX consortium, which is run by Francesco Montoni, I chair their advisory group, coupled with AVI via pharma with a different chemistry, morpholinos, which are much more stable than antisense oligonucleotides, and they also got very promising results by direct injection. And what they were doing was just skipping exon 51, which is applicable to 13% of patients. A systemic trial by going in subcutaneously has recently been completed by ProSensor in collaboration with GlaxoSmithKline. That's not published yet, but the presentations and the press releases, actually the press releases you should never believe because it's a biotechnology company. Well, you should take with a pinch of salt. But it's nevertheless very promising indeed. They don't have enough dystrophin. They're only getting about 20%, but it's at least a step in the right direction and a huge step in the right direction. So they're going through with phase 3 trials. And with the PMO, this systemic trial is still ongoing, but again, there are very favourable results. So why don't we just wait for that to come through? The problem is you'll have to put incredibly high doses of your PMO, your antisense oligonucleotide, and you have to deliver it systemically, and you have to keep on doing it. The other problem is it will only get to certain muscles and not to all muscles. For example, there's very little efficacy in the heart. So the current challenge really is to try and get either, first of all, better targeting, and I'm going to use viruses for that, and I'll show you some of the data that we have with that. And we also have to get one oligonucleotide that will address the deletions in all patients rather than have to go to the regulatory authorities and clinical trials for each type of mutation. So I'm going to tell you about how you can improve the PMO, and this was a collaboration with AVI Pharmaceuticals. And just to show how good this mouse is, I hope it'll work. No. Go backwards. No. Now then, that's better. So this is the double knockout mouse at about 3 weeks. And he's not going to move very much. He's just rummaging around in the corner, and he will fall over, as I said, in 20 weeks. This is the mouse we have treated with the PMO on a weekly basis. In fact, these mice are now 1.5 years old, and they are still running around as mischievously as any normal mouse. So if you do add a peptide tag to these morpholinos, you can not only target more muscle, but you can also get a much more dramatic effect than if you just use the antisense or the morpholino alone. And so the AVI are now going in for their next trial using this construct that they used in these experiments in collaboration with us. So we still have the problem that we have to be able to get high levels of dystrophin produced, and we still have the problem we have to repeat deliver. So one of the advantages of using adenovirus is that you can go in once and it's still there in 1 year's time. So we thought that if we could transiently immunosuppress, we might be able to take advantage and use AAV. We can also and I'll use a trick which is using the U7 SNRNP structure, put the antisense there, attached to this structure, which brings it close to the splicing machinery to induce more efficient skipping. So we do single intravenous injections, and this is just in the MDX mouse. This is where we started. That's an unskipped exon at the top line here. These are the PCR products of the skipped exon. You can see even in the protein, that relative to the wild type, we do get some full-length dystrophin. We don't get it in all tissues. In this particular one, we do get it in the heart, but it's nowhere near the wild type. Again, you can further manipulate the splicing machinery because you can use additional suppressor sequences. So what orally developed was not only the ability to put this into U7 vector, but also to have a little tail here, which means that you can recruit other suppressors which will induce this exon skipping even more efficiently. And I'll just show you that very quickly. You can see the difference here. There's the unskipped and there's the skip. So virtually all of that messenger RNA skips now the exon we want it to skip. And you can see here we also get then very close to wild type levels of the protein produced. So this can work extremely efficiently. So if we've got U7 and we've got one antisense oligonucleotide, the great advantage is that we can add lots of these oligonucleotides all together to think about addressing multi-exon skipping. As I said to you, a single one, if you skip exon 51, which is one of the most common ones, it's going to be 13% of patients. If you skip exon 44, it's going to be 7% of patients. So it's a very tedious way of getting through clinical trials to treat all patients. So if we could skip from 44 through to including 55, then we'd be able to tackle 63% of DMD patients. So that's the theory. And these, therefore, are all the oligonucleotides that we could optimize using this tag technique to get a efficient multi-exon skipping. And this just shows you again one of those exons, which used to be very difficult. If you use Orally's developed tag, we can get efficient skipping of even those difficult exons. So I'm going to show you lots of little... These are all of the constructs we've tried, and the reason I show you all of that is that, as you might expect, the nature does not splice things in the order you would expect. And we have no idea how the machinery in muscle puts all these exons together. It might splice exon 44, and then it might splice exon 48, or it might splice exon 47. So what we do have, and you can see that when you come to do the PCR again, this is... it does skip more than just what you want it to skip, and this is skipping 5 exons. So all I can tell you for the moment is that we have got a combination, but it's of 2 sets now, where we do have more than 5 exons in a single vector. So, and we can do it in, we can do it in mice. And so our preliminary results then make me feel that we may be able to do a single vector or perhaps 2 vectors, which would be possible to take into clinical trials. So a bit of reflection. I started this work in 1980. We didn't even know where the gene was. We knew it was on the X chromosome. The Duchenne muscular dystrophy gene was thought to be different from the milder form, Becker muscular dystrophy. One was on the short arm of the X chromosome, one was on the long. So the first thing we could do was to develop a prenatal diagnosis. As I said, Lou Kunkel cloned the gene. I'm embarrassed to say that it's taken a long time, from 1986 to 2010, for us to get really efficient, promising clinical trials. And that means good biology and good physiology. And in my life, I started off as a chemist, I then became professor of genetics, I then became the Dr. Lees professor of anatomy, and then I had to merge the physiology and anatomy departments in order to do my physiology. So we're all going in the right direction, but it's the basic science that both so much fun and so productive in this particular project, and it's been an international collaboration. And so for the very first time this year, I'm standing and saying, we have got clinical trials on viral delivery, which I think are going to be difficult to go forward. We've got very promising things for exon skipping, and I hope utrophin upregulation works. And I'll come back and celebrate with you if it does. So with that very message, and without forgetting that all the people in my lab past and present in bold, who've contributed to this work, Summit and Biomaran, who've taken this through to the clinic, the chemists, Angela Russell and Steve Davies, Steve Wilton and Sue Fletcher with AVI with that remarkable mouth, and we wouldn't be able to do it without the charities, the Medical Research Council, and of course all the patients that persuaded me this was an important disease to study in the first place in early 1980. Thank you very much.